All right, so in this video, we're going to be going over the first part of chapter 17, which is going to be on protein synthesis. And I can't stress enough how important this chapter is because um, the reality of it is that we talked about DNA and you know what DNA is, but without DNA, you can't make proteins. But kind of you need to think of proteins as being kind of like the final product. So really, this is going to be this portion of this video is going to just be kind of an overview of what protein synthesis is. The next one will be more detailed on how it actually occurs and the steps involved. So before we do anything, you do need to make sure you feel comfortable on what protein structures are. And this is a review of chapter 5, which was in the first unit. So just to remind you guys what are proteins. They're made up of tiny amino acids linked together. And they fold, and proteins can be a number of different things. We refer to it as a protein as once it actually has its functioning abilities. So remember, proteins can be things like enzymes. They can be involved in cell signaling. They can be a signal itself. It could be proteins are everything, really, when you think about it. So let's look at the monomer. So remember, each one of these, like glutamine, serine, each one of those is representative of one of these amino acids. Okay. Now, one thing that you want to make sure that you at least understand is what we call the backbone. So notice here, I have my amino, my NH2, my alpha carbon, and then my carboxyl. That's going to be the same for every single amino acid. It's the R group, that side chain is what can change. And this is what's going to give it its chemical properties. So that R group, that side chain, there's 20 different amino acids as shown here. And if you look, the backbone is all the same, the amino, the carbon, the carboxyl, the amino, carbon, carboxyl, okay? Now, sometimes it's drawn as NH2, sometimes it's NH3 with the plus sign. It's the same thing. So same thing with the carboxyl group. It could have the OH or it could just have the O minus. They're going to be the same. So just notice here, the R group, like glycine, I just have a hydrogen. Alanine, I have CH3. Leucine, I have this couple of carbon chains. It's the R group is how we identify amino acids because the backbone's all the same. So here, all of these R groups are going to be non-polar. Here you've got some polar R groups, and if you look at the R groups, it makes sense. Like this one has a hydroxide that's really, really polar. Same thing over here, really polar. I got an amino group that's polar. So then you got acids and bases. So understanding that, you don't have to memorize like lysine is going to be a basic amino acid. You just have to kind of understand that the R group is what gives each one of these its identity. And realistically, there's actually 22, but for the most part, we're going to assume, not assume, but biologically, at least in this class, we're going to be talking just about these 20. And how these things get rearranged can make literally any protein. So that's crazy. So you want to think of these almost as like, at least I think of them all like as Legos. There's only a couple of different types of Legos, but how you can rearrange them is really endless. Same thing with proteins. So it's how these things get rearranged. So one thing you want to look at is how proteins fold. And again, I have more detail in my chapter 5 video notes. Remember, the primary structure is just the order. And this is going to be determined by the DNA. So that's kind of be where this video is going to be starting, is you want to make sure that you understand the relationship between DNA and protein. So when you think about the primary structure, this just kind of revisits, right? It's a dehydration. You lose water. The bond that holds these things together is a peptide bond. And the biggest thing about what we're going to get into in this chapter is, well, what is the relationship between DNA and your protein that you're building? So the primary structure, remember the protein actually, there's really 4 steps involved in actually finalizing your protein. The primary structure is determined by the DNA. And realistically, this occurs at the ribosome where you're building your primary structure. And then it's going to start to fold. It folds the first time, what's called its secondary structure. Hydrogen bonds just within the backbone. And then it folds again, looking at the R groups. And so big things on this one is you want to remember, like non-polar R groups want to be next to non-polar R groups. Polar ones want to be next to polar ones. The disulfide bonds, and then your ionic bonds. So just understand kind of the main folding. So it folds in the backbone, that's your secondary structure. It folds again in the tertiary. And then this last one is the quaternary structure. That's where you have multiple polypeptides 1. So I'll call this peptide 1, peptide 2, peptide 3. But when these things are all three combined, this gives it the actual quote unquote protein. So like collagen is a protein that that has 3 polypeptides. All right, and that gives it its functioning capability. So with that being said, let's kind of introduce what chapter 17 is going to be all about. And you want to understand kind of this relationship of what DNA is. You know how DNA replicates, you know that it is containing genetic information, but this is kind of the bulk. Anything that is considered a living species really has to be able to do this. It has to be able to make proteins. Life without proteins cannot happen. So that info in the DNA, it's a specific sequence of nucleotides, right? So it's the A, the T, the C's, the G's, okay? That order is gonna determine the traits a particular species has. So this chapter, and really this unit is about gene expression. So if you don't know my definition of gene expression, it's really just controlling of protein synthesis. So when we think about gene expression, that word, we're going to talk about it a lot, but just make sure you understand what is gene expression. It's how a cell can control protein synthesis. How does it know which proteins is it making? How do we turn it off? And there's really 2 parts. There's transcription and translation. So this last kind of step is proteins are the links between genotype and phenotype, right? So when we do Mendelian genetics and you do these Punnett squares, when we think of the genotype, that's the genes that a cell or a species is carrying. The phenotype is the proteins that actually get expressed, okay? Now, don't think phenotype just as being something like, you know, blue eyes or hair color. Phenotypes could be something along the lines like actually making an enzyme. That could be an expression of the phenotype. Can it make an enzyme or can it not? Your AP test likes to kind of throw those questions in. So with that, let's kind of get into the basics here. I'll get a little specific later on protein synthesis in prokaryotes, but one thing that this video is going to focus more on is eukaryotic cells, so things with a nucleus. So this is known as central dogma. So you for sure want to make sure you know kind of this orientation. What is central dogma? It's this. DNA is read to make mRNA. mRNA is then read to make your protein. So if you look right here, let's turn on the little, well, you have your nucleus. You actually read a small section. You make your mRNA right here. mRNA is sent out to the ribosome, and then this is basically building your protein right here. So transcription and translation are going to be the 2 main components to this, okay? So remember, DNA to DNA, that's replication. That's chapter 16. Remember, cells only replicate DNA when they're dividing. But you need to understand that if a cell doesn't replicate the DNA correctly, it can't make that protein in the new cell. So let's just kind of get into the ins and outs of what this is going to be focused on. So DNA is read to make mRNA. That's transcription. If it's eukaryotic, it's taking place in the nucleus. Now the mRNA to the protein, that is going to be referred to as translation. That takes place at the ribosome, okay? Now, the one thing that I have to mention here is 1 gene and 1 protein hypothesis. So proteins are often composed of several polypeptides. That means multiple of these polypeptides linked together, okay? Now, 1 gene, 1 protein basically is going to look something a little different like this. So it's not like 1 gene is going to make 1 complete protein. You actually have multiple genes that make up a functioning protein. So this 1 gene, so if you look here at hemoglobin, notice that I have this polypeptide in orange, this one in green, this one in purple, this one in kind of like this pinkish color. So this protein in your red blood cell, there's 4 polypeptides. And it's not just 1 big gene that makes it all function. There's actually a couple of genes that help control it. So let me kind of show you what this looks like in this drawing I made up. So if you think of it from this standpoint, and if you see it from this and you understand it, you're in good shape. So let's just kind of remind you that if this is my chromosome, right? And notice that on this 1 chromosome, like if we actually zoomed in, it would be A's and T's, C's and G's, T's and A's. So I just kind of am highlighting this, a few things here. Notice I have more than 1 gene on a chromosome. That is absolutely valid. That is super correct. But what you need to understand is this. Let's just look quickly at this first gene, and fixate on what's going on here. So a gene, if you forgot, a gene is just a section of DNA that actually is read. When I say this section of DNA is read, that means I'm reading it to make my messenger RNA. So gene 1, we do transcription, meaning we make your messenger RNA. That messenger RNA is going to be translated at a ribosome to make this polypeptide, okay? On the same chromosome, I have a second gene called gene 2. I'm going to do transcription. I'm going to make my messenger RNA, okay? Now this gene makes this mRNA, which is different in the sense that my sequencing is different. Now this mRNA is going to be translated to make this polypeptide. So I have two genes. They each made their own individual polypeptide. Now here's what you need to think, is that this polypeptide by itself doesn't do much, if anything. This polypeptide by itself doesn't really do anything, but when you actually combine them, this is going to give that finalized protein, giving that protein its functioning capabilities. So when you think of that one gene, one protein, hemoglobin is a good example of that, where you have a few genes that actually are going to be associated with building that complete hemoglobin protein. So, but understanding that it makes that final protein, it's multiple polypeptides combined. So that's just basically what we want to kind of get out. Nice little recap of what's a gene and the purpose. Now, in terms of that, let's kind of emphasize this. RNA is kind of being the bridge between the genes and the proteins, right? So the mRNA is your messenger mRNA. So we're going to get into what's transcription and what's translation. Now, just to kind of give you the brief overview, the next videos will be a lot more specific, but remember, this is really, really good in terms of what they're showing here. We have a big, long DNA molecule here, a big, long chromosome. I'm only reading one small section. Remember, a section of DNA that actually gets read is called a gene. So you wouldn't actually read an entire chromosome because a lot of our DNA doesn't code for anything. So if you look here, this gene right here, I have some A's, some T's. I only read one side, so I'm going to actually read it. Now let's go through the basics here. Remember, DNA is your double helix. So there's two strands running anti-parallel. I'm only reading one side of my DNA. So this A is going to pair with a U. Remember, in RNA, there's no thymine. It's uracil instead. Cytosine pairs with the guanine. Cytosine pairs with the guanine. And so remember, in transcription, that's when you make your mRNA. In translation, that's when you read the mRNA to build your proteins. So remember, you need to think that this at the end is your end goal. Your protein is your end goal. How does it get there? Well, you need to make your mRNA. How did your mRNA get the right sequence? Well, you're reading the DNA. So that's the relationship between DNA and your protein. So cannot stress that enough and how important that is, because what we see in everything is really the proteins that get expressed. So let's kind of remind you of the basics between DNA and RNA. So this is showing you obviously the double helix running anti-parallel. Here's your bases, C, G, A, and T. Now, RNA stands for ribonucleic acids. Now the bases are going to be C, G, A, and we have uracil, no thymine as one of the bases, right? But there are a few other differences as well on DNA versus RNA. And one thing is the sugar, which it's not super difficult, but I'll point it out now. DNA stands for deoxyribose, RNA stands for ribose. So basically, if you looked at DNA, if you had a phosphate and you had a sugar, so let me redraw this real quick. So if I had a phosphate and I had a carbon here, here's my carbon. I'm building my sugar right here, carbon, carbon, carbon. And then you have your base. So whether that be an A, T, C, G, or U, all right? Now on my second carbon, I have a hydroxide. Now, if I number these, carbons 1, carbon 2, carbon 3, carbon 4, carbon 5, I have an OH here. Now, really what we need to fixate on is this hydroxide group right here, okay? If I have this hydroxide group, I would call it RNA, ribonucleic acid. Now, if all of a sudden, I shouldn't say all of a sudden, but without that hydroxide group, it's that second carbon that this would actually be a, because remember, what's a nucleotide? It's your phosphate, your sugar, your base. This would be an RNA nucleotide. Without this hydroxide, it would essentially be a deoxyribonucleic acid. So it's the difference of that hydroxide, okay? Now, Regarding that, I'm going to basically end the video here because this is just an overview. The next video will be more specific on transcription.